Welcome to the Bioscan Hub channel. Hey, if you're into hematology and lab science, do us a favor and hit that subscribe button. It really helps us out. Okay, so today, we're going beyond just saying, yep, that's anemia. We're going to get into the real detective work. We're talking about the specialized tests you'll use to figure out the why behind hemolytic anemia. This is where the real skill comes in. Ready? Let's get started.
So here's our game plan. First, we'll set the scene, the main puzzle. Then we're diving into 3 key areas. We'll look at the cell's engine, its basic structure, and how it actually does its main job, carrying oxygen. And then, the most important part, we'll connect all those lab results back to what it means for the patient.
All right, so imagine this. The first round of tests are back. You've confirmed that a patient's red blood cells are being destroyed prematurely. Okay, great. That's hemolytic anemia. But that's just the tip of the iceberg. Now the real work begins, the part where your expertise as a lab professional is absolutely critical. We have to find out why it's happening. And here's the tricky part. A lot of these patients, they don't even feel sick. They might have no clinical symptoms at all. And that's exactly why the lab is so important. You're the one looking under the hood. Your analysis is what's going to uncover that hidden truth. Is it some inherited enzyme deficiency, a problem with the hemoglobin itself, or maybe something they were exposed to? That's what you're going to find out.
Okay, first up, let's take a look inside the red blood cell's engine room. We're talking about its enzyme pathways. Think about it. These are what generate all the energy that cell needs to just live. If that engine sputters and fails, well, the whole cell's going down.
So the first big player we need to talk about is an enzyme called Pyruvate Kinase, or PK for short. Now, a deficiency in this guy is actually the most common reason we see hemolytic anemia related to these energy pathways. You can think of ATP as the cell's battery. If you have low PK, you get low ATP. The battery's dead. And without power, the cell's membrane just falls apart, gets unstable, and the cell is destroyed way too early.
So when you get a low PK result back, it really points to 2 main possibilities. It could be congenital, something the person was born with, or, and this is really important, it can be acquired, meaning it's a side effect of another serious disease, like myelodysplastic disorders or even acute leukemia. And making that call is huge, because if it's acquired, you treat the main disease, and guess what? The PK levels can go right back to normal.
All right, next up is a big one, glucose-6-phosphate dehydrogenase deficiency, or G6PD. You absolutely have to know this one. It's a sex-linked disorder, so you're going to see it mostly in males. And for the most part, people with it are totally fine, no symptoms. But when they're exposed to some kind of oxidative stress, we're talking certain drugs, a bad infection, or famously, even eating fava beans, boom, they can have a sudden, really severe hemolytic crisis. And this whole process is tied to something called Heinz bodies, which we're definitely going to get into in a bit.
Now, this is why paying attention to a patient's ethnicity and background is so important. See, G6PD deficiency isn't just one thing. There are different versions, different genetic variants, and they're way more common in certain populations. Knowing this can seriously help you narrow things down. Like, if you have a male patient of Southeast Asian descent, the Mahedol variant should be high on your list of possibilities.
Interpreting G6PD levels is a bit of a puzzle. A low level, okay, that points you straight towards G6PD deficiency. But what if the level is high? That's a clue for something else entirely. It could be pointing to things like untreated megaloblastic anemia or even hyperthyroidism. It's just a perfect reminder that you can never look at one lab result in a vacuum. It's always about the whole clinical picture.
Okay, pay close attention to this. This is a huge clinical alert. The Mediterranean variant of G6PD, it is especially severe. For these folks, eating fava beans isn't a small thing. It can trigger a massive, and I mean potentially fatal, hemolytic crisis. This is where your speed and your accuracy in the lab can literally save a life. It's that serious.
All right, so we've been looking at the engine inside the cell. Now let's switch gears and look at the cell's bodywork, its physical structure. How tough is this thing, really? Is it built to last that chaotic trip through the bloodstream, or is it super fragile and just waiting to fall apart?
And the way we test that is with the Osmotic fragility test. The whole idea is actually pretty simple. We take red blood cells and we put them in salt solutions that get weaker and weaker. And we just watch to see when they pop. Think about it. A normal red blood cell, that donut shape, has some give. It can swell up a bit. But a spherocyte, that's a cell that's already stretched tight and round, like a balloon that's been blown up as much as it can. It has zero room to expand, so it bursts really easily. That's what we call increased fragility. So let's break down what the results mean. If you see increased fragility, cells popping easily, that's a classic sign of something like hereditary spherocytosis. But what about the opposite, decreased fragility? That's when the cells are actually tougher than normal. You see this in conditions that make thin, flat cells, like an iron deficiency anemia or thalassemias. They're like half-empty water balloons. They can soak up a ton of water before they even think about bursting.
Okay, let's circle back to those Heinz bodies we mentioned. Just to be clear, these aren't some kind of bacteria or parasite. They're literally just clumps of junk, damaged, broken down hemoglobin. When hemoglobin gets slammed by oxidative stress, like in that G6PD crisis we talked about, it basically falls apart and gets all sticky, clumping up on the inside of the red cell membrane. And those clumps, those are Heinz bodies.
So how do you actually spot these things? Well, your standard blood stain isn't going to cut it. You need a special technique called a supervital stain. It stains the cells while they're still alive, which is key. And when you're looking down that microscope, it's so important you can tell them apart from other stuff. So here's your key takeaway. Heinz bodies look like little pale blue dots, kind of pushed off to the side of the cell. Don't confuse them with the dark purple stuff you see in reticulocytes. That's just RNA.
All right, for our last set of tests, we're looking at something a little different. This isn't about power or construction. This is about the red blood cell's #1 job, dropping off oxygen where it's needed. And to do that, it relies on this tiny but incredibly powerful little molecule called 2,3-diphosphoglycerate, or 2,3-DPG.
So what does 2,3-DPG do? Well, you can think of it as the key that unlocks oxygen from hemoglobin. It basically convinces hemoglobin to let go of the oxygen it's carrying. So if your body senses it's not getting enough O2, maybe because of anemia or you're up on a mountain, it cranks up production of 2,3-DPG to make sure your tissues get every last drop of oxygen they can.
And this right here leads to a massively important clinical situation. Think about blood in a blood bank. The longer it sits there, the more its 2,3-DPG levels drop. Now imagine you give that older blood to a really sick patient. Those transfused red cells, they're full of oxygen, but they're oxygen hoarders. They won't release it to the tissues that desperately need it. The patient's pulse oximeter might look perfect, but on a cellular level, they could be starving for oxygen. This is why understanding 2,3-DPG is absolutely essential in transfusion medicine.
Okay, let's pull all these threads together now. We've talked about the cell's engine, its structure, how it delivers oxygen. Now let's see how all the incredible work you do in the lab actually changes how a patient is treated.
Now, before you can even run these tests, a few things have to happen right. Of course, talking to the patient is #1. But from our perspective in the lab, the absolute most critical thing is this: no blood transfusions before you collect the sample. Just think about it. If you're testing for fragile cells and the patient just got a bag of super healthy donor cells, well, your test is going to look totally normal and you'll completely miss the real problem. It'll give you a false negative. And once your results are out, they directly guide what happens next. If you diagnose G6PD deficiency, the biggest thing is education. Here's a list of drugs and foods. Do not touch these. If you find severe hereditary spherocytosis, the treatment might be surgery, a splenectomy, to remove the very organ that's been chewing up all those fragile red cells.
And this is such a cool way to see what the spleen actually does. It's the body's ultimate quality control inspector. It's constantly pulling out damaged cells, like the ones with Heinz bodies. So what happens when you take it out? Well, all those defective cells just keep on circulating. And you can get this wild result where you look at their blood and find Heinz bodies in more than half of their red blood cells. It's pretty dramatic.
So at the end of the day, I really want you to remember this. Never, ever underestimate how important your job is. You're the one connecting all the dots. Your analysis is what provides the hard evidence, the data that links a patient's entire condition to a tiny specific problem inside a red blood cell. That's the power you have in the clinical lab. So I'll leave you with this to think about. After everything we've gone over today, the enzymes, the cell structure, all of it, if you had to pick just one of these tests, which one do you think tells you the most about how well a red blood cell can handle stress, and more importantly, why? Think about it, because that's the kind of critical thinking you'll be doing every single day. Thanks for joining me.